Chapter 12 is all about the cell cycle. We're going to be focusing on how cells are able to divide and duplicate. And this goes back to one of those hallmarks of characteristics of living things. Being able to reproduce independently is one of those hallmark characteristics that separates living things from non-living matter. And we are able to do that by reproducing cells, which is also known as cell division. When you have a unicellular organism, because it consists of a single cell, when that cell reproduces, it is reproducing the entire organism. Multicellular organisms will reproduce cells to be able to grow from a fertilized cell, such as an egg, to be able to grow and also to be able to repair. And so not every cell is necessarily going to go all the way through the division process, but cells need to be capable of undergoing division when needed for that organism to be able to survive and reproduce. So the majority of cell divisions are going to result in identical daughter cells, specifically with the chromosomes present in the nuclei. Cell division is going to consist of both mitosis and cytokinesis. Meiosis would be the notable exception to that, where the DNA is actually, or the chromosomes are divided such that each reproductive cell, each gamete, would have only one full set of chromosomes, and it would need another gamete in most situations to come along to get its second set of chromosomes. All of the DNA that's present in the cell is going to constitute that cell's genome. Remember that DNA is not just in the nucleus, it is also found in your mitochondria, and it can be found in your chloroplast as well. The genomes can either be one large DNA molecule, which is what we tend to see in our prokaryotic cells, or you can have lots of individual DNA molecules, which is what we're more likely to find in eukaryotic cells. And again, the DNA gets packaged into units known as chromosomes. These chromosomes, specifically the eukaryotic ones, are made of chromatin, which is not just DNA, it's also a protein, and they condense during cell division so that the individual chromosomes can be seen. The different eukaryotic species have a different number of chromosomes in their nuclei. They're somatic cells. All the cells besides the germ cells are going to have two sets of those chromosomes, while the gametes, your reproductive cells, sperm and eggs, will only have one full set of chromosomes. They'll always have half the number of chromosomes that are found in your somatic cells. So in cell division, the first things that need to take place are the replication of your DNA and being able to visualize the chromosomes, having them condense. When the DNA is replicated, the replicated chromosomes stay together. They are actually joined at the centromere, and they will consist then of two sister chromatids, and they undergo separation as a part of cell division. Again, where they connect at is a centromere, and that's where they're most likely to be attached. They also can be attached along their arms. We'll talk about that a little bit more later on. As a result of cell division, these sister chromatids separate and move into two separate nuclei. And once they have separated, they are no longer considered chromatids. They are now considered individual chromosomes. So there's your chromosomes. You've got the duplication into the sister chromatids. Again, they are attached at that centromere, but they can also be attached by cohesions along the chromosomes' arms. And then when the sister chromatids are separated in mitosis, and they result in the formation of two new cells, they are back to being chromosomes. So mitosis is focusing on dividing the genetic material present in the nucleus, while cytokinesis is division of your cytoplasm. And so gametes are going to use division to be formed, but because their chromosome number needs to be halved, they actually go through a second series of events, and that's known as meiosis. We'll spend more time on meiosis in our next chapter. In meiosis, you will see that there are some events that take place that do not take place in mitosis, which allows each individual cell to not necessarily be identical to other daughter cells. And again, they only have half the number of chromosomes that their parent cells would have. Mitosis alternates with interphase as part of the cell cycle. You have the mitosis, the M phase, and then you have interphase taking place, which is what the majority of the cell cycle is going to be. This is when your chromosomes get duplicated. This is when the cell grows and is getting ready to undergo that division process. Initially, after coming out of mitosis, your cells that are in interphase are known to be in the G1 phase, the first gap. If they pass various checkpoints, they can move into the S phase where the DNA is duplicated, and then they can continue to grow in the G2 phase. And once they pass certain checkpoints, they can move into mitosis. So the cells are growing throughout interphase, but only the chromosomes get duplicated during the S phase. And so there you can kind of see time-wise that you spend very little time in mitosis compared to interphase. So mitosis itself is divided into four main phases: prophase, metaphase, anaphase, and telophase. Pro-metaphase has kind of gone in between prophase and metaphase because that is one of the times that you're going to have, or one of the phases the cells are going to be in longer during mitosis, and it's kind of a unique blend of prophase and metaphase. Cytokinesis does not occur separately from mitosis. It actually overlaps a little bit of anaphase, but primarily telophase. So in interphase, your nucleus is still surrounded by a nuclear envelope, and there is either one or more nucleoli present in the nucleus. In animal cells, there will be centrosomes present that have duplicated, and the chromosomes that have already been duplicated are there, but you cannot visibly see them. They still are in the form of chromatin. The centrosomes start to assemble microtubules, and that's one of the names for centrosomes, the MTOC, Microtubule Organizing Center, which are going to help with separating the chromosomes and pulling them into new cells. And it'll also help expand the cell to make it easier for the cell to divide and be of a decent size. During prophase, the chromatin fibers condense, they become more tightly coiled, and you can actually see the chromosomes in the form of sister chromatids. You no longer see your nucleoli. And again, I told you depending on your species, some of the arms of the chromosomes may be attached to one another via cohesion proteins. You will notice that part of the centrosome, you can see some microtubules expanding. So you have that early mitotic spindle. You have the aster forming, which are the shortened microtubule ends that are kind of separate. They're radiating from the centrosome. And you will see the centrosome start to move to opposite ends of the cell to better be ready to separate into two daughter cells. So a little bit more about that mitotic spindle. It is basically a just made of microtubules, and they are able to grab onto chromosomes at the centromeres, and they are able to help to extend the cell to make it larger and better prepared for that division process. I talked about the aster, how it's basically just shortened microtubules that are coming off of the centrosome. So during prometaphase, the nuclear envelope starts to break down, and you will notice that microtubules are starting to enter into what was the nucleus. You can more visibly see the chromosomes, and you can see that at the centromeres on the chromosomes, there's now this protein complex known as a kinetochore. Specific microtubules will grab on to those kinetochores so that they are better able to separate the sister chromatids as the mitosis process continues. The non-kinetochore microtubules are able to work across the nucleus and around the nucleus to interact with other non-kinetochore microtubules at the other centrosome, now that the centrosomes have moved to opposite ends of the cell. Again, just helping the cell to expand. And so what we have learned over time is that when these kinetochore microtubules start to pull back and separate the sister chromatids from one another, they do start to break down, and they actually are breaking down on the end that is connected to the chromosomes, so the kinetochore ends. At metaphase, your centrosomes are going to be at the two opposite ends. They're going to be 180 degrees roughly apart from one another, and the chromosomes will be lined up parallel to them in the center. Basically, it's at this invisible midway point between the two poles that the centrosomes consist of. Before the cell can move past metaphase, kinetochore microtubules have to be attached to each of the centrosomes or the kinetochores that are found on the centrosomes of these chromosomes. And there will be kinetochore microtubules, one from one spindle pole, one from the opposite spindle pole, attached to each of the sister chromatids' kinetochores. All right, so there you can see more visibly and I was calling it centrosomes. I'm really sorry. I was going for centromeres earlier. The centromeres are what the kinetochore microtubules are trying to reach out for because that's where the kinetochores are found. And the centrosome is extending those microtubules out, both the kinetochore and the non-kinetochore microtubules, to make sure that each chromosome is attached to a kinetochore microtubule, and that for the sister chromatids, that the two chromosomes each have a kinetochore microtubule coming from the opposite centrosome, and then you have the overlapping non-kinetochore microtubules from both sets of centrosomes. When we reach anaphase, we do see the sister chromatids separate. If cohesions have been involved, they are cleaved. We see that they split at those at the centromeres, at the kinetochores. And so the kinetochore microtubules start to draw in those separated chromosomes to opposite ends of the cell. They get shorter, they depolymerize at the end that's connected to the chromosome as opposed to the end coming out of the mitotic spindle. Your non-kinetochore microtubules are continuing to grow. They are actually overlapping, pushing up against each other, causing the cell to get longer to better be prepared to divide with the cytoplasm. And at this point, you will have more of the chromosomes towards that mitotic spindle than you will have towards the center of the cell. During telophase, you have your chromosomes pretty much in your two separate cells. We have the nuclear envelopes start to form again. The nucleoli show back up. The chromosomes start to become less condensed. If there are microtubules that have not depolymerized, you will see them finish up that depolymerization process. And this is also where cytokinesis is going to start to take place, the division of your cytoplasm, and mitosis is going to be complete at this point. So again, I said cytokinesis is going to happen partly in anaphase, but also in telophase. It's going to be when your cytoplasm divides. It's going to be really hard to divide your cytoplasm evenly, but the goal is to get, each cell the resources it needs to be able to survive and thrive. In animal cells, cytokinesis occurs via cleavage. The cell gets pinched in the center. In plant cells, you will have various macromolecules come to the center and form a cell plate. So there's that cleavage furrow that we see with an animal cell that's basically going to be formed by a ring of microfilaments. Whereas with the plant cells, you've got vesicles that are coming together to form that cell plate. And after the cell plate is formed, the cell wall can be generated. With bacteria, you are going to reproduce a little bit differently. They're going to use binary fission because they basically are just going to be having that single chromosome. We see this both with the bacteria and archaea domains, the prokaryotes there. The chromosome is replicated starting at its origin of replication, and then the two chromosomes basically are growing further apart from one another. And then the plasma membrane basically pinches inward to divide it into two separate cells. So there's kind of an example of what is taking place with that bacterial chromosome in terms of getting it copied. Once the DNA has been copied, the membrane can pinch off and you have two daughter cells. So it is thought that mitosis is most likely evolved from this binary fission process. You can see there with bacteria, with the dinoflagellates, you have a little bit more you have a more structured nuclear envelope. You've got microtubules taking place. As we get further along with protists, we can see we have kinetochore microtubules. We have non-kinetochore microtubules. We still have that nuclear envelope. And then as we get into eukaryotes, we have the nuclear envelope being separated. We have the microtubule organizing centers. And we definitely have many more kinetochore microtubules as well as non-kinetochore microtubules working to divide the cell properly. So this cell cycle is regulated by a control system. Cell division is not constantly happening with every type of cell. It depends on what type of regulation is taking place and what's going on with the cells. Cancer cells are able to escape this system or in generally speaking, and that can cause a whole host of problems. The cycle appears to be driven by signals in the cytoplasm, and there's some data to support this, where cells that were at different phases in the cell cycle were fused together. So here we have in this first experiment, we have a cell that's in S phase that is going through division, and then a cell that was in G1 that was not duplicating its DNA. It was just growing. When they two were fused together, it caused the cell that was in G1 to start to go through that duplication of DNA. In experiment 2, we took a cell that was in mitosis and we fused it with a cell that was in G1. When the cell that was in mitosis was fused with that cell in G1, it caused the G1 cell to also undergo mitosis prior to it going through the S phase, so it did not have its DNA duplicated. So there's just some data to kind of support that there's some chemical signals in the cytoplasm of cells undergoing certain phases that can cause cells that are not in those particular aspects of the cell cycle to start to go through them. And so this control system is similar to a clock. You have both internal and external signals. There are specific checkpoints the cell has to pass before the cell cycle can continue. And for many, many cells, it's very difficult for them to move out of that G1 phase into the S, into the division of the duplication of your DNA. If they do not get that checkpoint go-ahead signal, they actually move into an inactive, non-dividing state known as the G sub 0 phase. But if the cell does receive a signal to continue on and to go through the synthesis of its DNA to duplicate it, continue to grow G2, it will eventually be likely to go through mitosis and divide. So we've got a checkpoint there with the G1. We also have a checkpoint before we go into mitosis, and then we have checkpoints during mitosis. We're not going to hit all of the checkpoints. We're going to be focusing more on big picture. But the main sets of proteins that help to determine whether a cell is going to be able to move out of G2 into mitosis are going to be based on cyclins. You have cyclins and you have cyclin-dependent kinases. Their activity is going to fluctuate based on the concentration of cyclin that is present in the cell, and that concentration increases as the cell continues to grow. It goes through G1, S, and G2. When the cyclins bind to the cyclin-dependent kinase, MPF, maturation promoting factor, is formed. And when there's enough of that MPF present, the cell is capable of moving past G2 and going into mitosis. So you need to have enough MPF to pass that checkpoint. So you can kind of see here that during G1, during S, during G2, during G1 and S, you don't see a whole lot of change in MPF, but as it starts to go through G2, you see the concentrations of cyclin increasing, and then eventually, once it gets to a level enough point, you see the MPF activity increase, and mitosis then can take place. So CDK is the kinase that cyclin is able to bind to form MPF, and CDK is recycled. As the cell goes through mitosis, the cyclin is degraded, the concentrations of it decrease again. So CDK is there all along. It's just whether or not there's sufficient enough cyclin to allow the cell to move out of the G2 phase and into mitosis. So you don't really start to see cyclin build up until you have moved through the duplication of your DNA and you've gone through G2. But once you have enough of that present, that can set off the checkpoint to move into mitosis. So there are other signals that can play a role in determining whether a cell is able to continue with the cell cycle. We've talked about those kinetochore microtubules, how the kinetochore complexes that are on the centromeres of your chromosomes have to get attached to spindle microtubules, the kinetochore microtubules, and if they are not, that can send a signal to delay anaphase, that the cells are not ready yet to properly divide. You could have growth factors. They can be secreted by specific cells that will allow the cell to continue to divide. Platelet-derived growth factor, PDGF, is able to do this in a culture setting with human fibroblast cells. Some other external signals, cells will typically only divide until they reach a point where they have occupied the available space. So if you have cells that are covering a petri dish, they are unlikely to continue to grow. The crowding of cells will reduce division of those cells. There's obviously plenty there. And then another factor that can play a role specifically with animal cells is anchorage dependence. The cells need to be attached to something, some sort of substratum, to be able to undergo that division. Cancer cells don't have to meet either of those requirements. They will keep dividing no matter how crowded they are, and they will divide whether or not they are attached to a substratum. And so in these next pictures, you're going to see kind of an example of that. You've got the anchorage dependence. The cells are anchored and they're able to divide until they fill up that base. They have a single layer. If you take some of those cells out, they will continue to divide again until they fill up the plate. And that's what we expect to find with our non-cancerous cells. That they want to be in contact with one another, but they don't want to be overly crowded, and they will grow based on their nutritional needs. And they have to be attached to something to be able to grow, and it also impacts on how dense they can be. With cancerous cells, you will notice that they are not just a single layer, that they form clumps of cells. They are not dependent on anchorage. You have cells that are growing that are not attached to any sort of base. And because they are not density dependent, that does not stop them from growing. They are able to grow in clumps. So Cancer cells typically are not going to respond well to these different control mechanisms. They don't necessarily have to have growth cells to be able to grow and divide. They can make their own, or they may be able to signal that growth factor without actually having that growth factor present, or they could have problems with their cell control system or their cell cycle control system. When a normal cell is converted to a cancerous cell, it is transformed. Your immune system is actively working to identify cells that are not functioning properly. But if it is unable to do so, you can have tumors form, which would be abnormal cells clumped together in otherwise normal tissue. And if they happen to only be at the site that they first started to become abnormal, you typically have what's called a benign tumor. Malignant tumors are ones whose cells are duplicating abnormally and have taken over adjacent tissues or have been able to metastasize, move to other parts of the body and form additional tumors. And so a tumor can grow from a single cell. Once that cell is growing out of control, if the immune system has not been able to take care of it, it can continue to grow and take over nearby tissue. Those cells can then move through your extracellular fluid, through your blood vessels, through your lymph system, into other areas of the body, and then new tumors can be established and able to grow and set off another host of issues.